Chemical classification of hormones. There are 4 classes: amines, polypeptides/proteins, glycoproteins, and steroids. Steroids are derived from cholesterol. Examples are testosterone, estradiol, cortisol. Glycoproteins are proteins attached to carbohydrate groups. An example is FASH, or luteinizing hormone. Polypeptides or proteins are chains of amino acids. And these are insulin or antidiuretic hormone. Amines are derived from tyrosine or tryptophan. And amines are modified amino acids. For example, an amine is epinephrine, thyroxine, or melatonin. Similarities between endocrine and neural regulation. Responses in the nervous system are immediate but short-acting. Endocrine responses are slower but longer-acting. Similarities in signaling. Receptor proteins are required for a cell to respond to a signal. That's number 1. Number 2, binding of a regulatory molecule to the receptor causes a specific sequence of changes in the target cell. Number 3, the action of the regulatory molecule must be able to be turned off or canceled. Endocrine glands and hormones. These glands are ductless and secrete their product into the blood. They secrete hormones which bind to receptor proteins on target cells, and they primarily compose organs that play major functions in other systems. Contraction of smooth muscle. Calcium is important, but plays a different role due to the lack of troponin. Smooth muscle responds to neurotransmitter, hormone, or action potentials. The steps are as follows. Number 1, the hormone/neurotransmitter binds to a G protein coupled receptor. Number 2, the alpha subunit will bind GTP and then interacts with a calcium channel. Number 3, the calcium channel opens and calcium will flood into the cell. Number 4, calcium will bind to calmodulin and activate it. Number 5, calmodulin then binds to myosin kinase and activates that. Myosin kinase will transfer a phosphate group to myosin, and this phosphorylated myosin can then bind with actin and begin the contractile process. Contraction in smooth muscle occurs more slowly than in skeletal muscle. To cause relaxation, myosin phosphatase must take place. Myosin phosphatase removes phosphate from myosin, and this will lead to relaxation. Relaxation can occur slowly and maintain tension for some time. Multi-unit smooth muscle. Individual cells must be stimulated by nerve fibers, whereas in single-unit smooth muscle, the cells are joined by gap junctions, and stimulation of some cells will cause others to contract. It's kind of like a wave effect. Some cells may function as pacemakers and then can contract on their own. Smooth muscle is smaller than skeletal muscle fibers. The actin filaments are very long to allow for excessive stretching, and there are no Z discs. Instead, dense bodies play a similar role. Cross-extensor reflex. This reflex ensures the other side of the body responds and prevents loss of coordination/balance. This involves double reciprocal innervation. On one side of the body, the flexor will contract and the extensor will relax to withdraw the foot. And this is due to the extensor being innervated by an inhibitory motor neuron, and the flexor being innervated by an excitatory motor neuron. On the other side of the body, the extensor will contract and the flexor will relax to support the weight of the other foot lifting. In this instance, the extensor is excited by a motor neuron, and the flexor is innervated by an inhibitory motor neuron. This is an example of a polysynaptic reflex because there are interneurons that are exciting or inhibiting the motor neurons, and they are in between the sensory and the motor neuron. The withdrawal reflex is the rapid response to pain. The steps are: number 1, pain receptors detect a painful stimulus. Number 2, the sensory neuron will conduct an action potential to the spinal cord. Number 3, the sensory neuron will synapse with excitatory interneurons that synapse with alpha motor neurons. Number 4, excitation in the alpha motor neuron will result in contraction of the flexor muscles and withdrawal of the limb from the painful stimulus. Reciprocal innervation. An example of this is when activating the stretch reflex in quadriceps. This will cause the quadriceps to flex and the hamstrings to relax. The steps are: number 1, the muscle stretch will activate the spindle apparatus. The agonist muscle, aka the extensor muscle, will contract in response to the stretch reflex. It is innervated by an excitatory motor neuron, and so that will cause the extensor muscles to contract. And now on the opposite side, the flexor muscles will relax because the sensory neuron is attached to 2 motor neurons. One of them is excitatory and one is inhibited by an interneuron. Due to the interneuron secreting inhibitory neurotransmitter, this will make the motor neuron to the flexor muscles inhibited, and this will cause the flexor muscles to relax. So you have the extensor muscles contracting, the flexor muscles relaxing. And this is known as reciprocal innervation. The Golgi tendon reflex. This prevents excessive stretching of tendons. It requires the Golgi tendon organ as a sensory receptor, and this is found adjacent to collagen fibers in a tendon on the ends of the muscle. Intense stretch stimulates action potential generation by these organs, which send signals to the spinal cord and synapse with interneurons. The interneurons then secrete inhibitory neurotransmitters, which block the activity of motor neurons controlling the muscle in question, and this leads to relaxation. And this is why you may drop a heavy weight after straining to lift it. So basically, there's too much stretching near the tendons, and the Golgi tendon organ will sense this, send a response to the spinal cord by way of a sensory neuron, and the sensory neuron will stimulate the interneuron will inhibit the motor neuron with inhibitory neurotransmitters, and then therefore tension on the tendon is reduced because the motor neuron is inhibited. And cannot excite the muscle. It causes the muscle to relax when it is inhibited, and that's to prevent excessive stretching of tendons. One more time, the steps are: number 1, tension on the tendon activates the sensory neuron. The sensory neuron will stimulate. Number 2, the sensory neuron will stimulate the interneuron. Number 3, the interneuron inhibits the motor neuron. And number 4, tension on the tendon is reduced. The Golgi tendon organ is it senses the stretch because it's the sensory receptor. Okay, the stretch reflex. This is also known as the knee jerk reflex, and it keeps quad muscles from overstretching as well as maintaining balance. The stretch reflex requires the muscle spindle as a sensory receptor, And unlike the Golgi tendon organ, which is only found near the tendon, next to collagen fibers, or just next to collagen, adjacent to collagen fibers, the muscle spindle is found like all throughout the muscle. It's just wrapped around fibers there, and the muscle spindle is the sensory receptor in this case. The sensory neurons innervate the spindle and generate action potentials when the muscle and spindle are stretched. These neurons directly communicate with motor neurons in the spinal cord, which innervate the muscle containing the spindle. And this is a monosynaptic reflex because there is no interneuron in the stretch reflex. It simply goes from the sensory neuron and that attaches to the motor neuron. Shortened stretch stimulates sensory neurons, which stimulate motor neurons in the spinal cord, resulting in rapid muscle contraction. The steps on the graphic are: number 1, striking the patellar ligament stretches the tendon and quadriceps femoris. Number 2, the spindle is stretched, activating the sensory neuron. Number 3, the sensory neuron activates the alpha motor neuron. Number 4, the alpha motor neuron stimulates extrafusal muscle fibers to contract. Okay. Polysynaptic reflexes. These involve interneurons, there's more than one synapse, and this includes at least one interneuron, and it may include more than one motor neuron. The monosynaptic reflex is one synapse between two neurons, a sensory and a motor neuron, which directly synapse together. Reflexes. While neurons are the basic structural unit of the nervous system, the basic functional unit is the reflex arc. The reflex arc is the simplest portion of the nervous system capable of receiving a stimulus and producing a response. It consists of number 1, sensory receptor, 2, a sensory neuron, 3, an interneuron, but not always, 4, a motor neuron, and 5, an effector organ. A reflex is known as an automatic response to a stimulus produced by a reflex arc, and it may or may not involve the brain. Reflexes are homeostatic, and they can maintain blood pressure and carbon dioxide levels, as well as prevent damage to the body. For example, the withdrawal response, or the withdrawal reflex. That is how it can prevent damage. The withdrawal reflex. Central fatigue involves a possible reduced ability of neurons in the brain to signal to neurons in the spinal cord to produce movements. This often occurs before muscle fatigue sets in. Muscle fatigue. Fatigue is described as the decreased capacity to do work and reduced efficiency of performance. Muscular fatigue may be caused by excessive potassium levels in extracellular fluid, depletion of glycogen, calcium accumulation in the cytoplasm, excess phosphate in the cytoplasm, and lowered pH from lactate production. Slow twitch versus fast twitch. Slow twitch is also known as type 1. This slow twitch contracts relatively slowly, while fast contracts rapidly. Slow twitch has a higher blood supply and fatigues less easily. Fast twitch has a lower blood supply and fatigues more easily. There are more mitochondria in the slow twitch fibers, and there are less mitochondria in the fast twitch fibers. There is a high level of myoglobin in slow twitch fibers and a low level of myoglobin in fast. Myosin heads break down ATP slowly in slow twitch fibers, and myosin heads break down ATP rapidly in fast twitch fibers. Slow twitch fibers are best for posture maintenance and endurance, while fast twitch fibers are best for rapid, intense movement. Oxygen debt and recovery / oxygen consumption. After exercise, you continue to breathe heavily, and this excess oxygen helps to repay the oxygen debt. Oxygen is stored by hemoglobin in the blood and myoglobin in muscles. Oxygen is used in metabolizing lactic acid by the liver. Phosphocreatine. This is synthesized during resting conditions, and it's useful for high-intensity exercise, but not endurance. Phosphocreatine is easily converted to ATP by creatine kinase, and it can be synthesized by the liver or a kidney or obtained through diet. Fermentation. This generates ATP by metabolizing glucose without oxygen. This occurs above the lactate threshold, and ATP is produced quickly but inefficiently. Lactic acid is produced and metabolized in the liver for additional energy, and the use of glycogen can power moderate and extreme / heavy exercise, but the amount of stored glycogen helps determine the length of exercise possible. Glucose uptake during exercise. During low-intensity exercise, about 65 watts of glucose is uptook. During medium, 130 watts, and during high intensity, 200. So the more intense your exercise, the more glucose that will be taken up. Aerobic respiration. This is the metabolism of glucose / fatty acids in the presence of oxygen. This is the dominant mode of metabolism during light exercise. And extra oxygen is stored by myoglobin and skeletal muscle. Increases in GLUT transporters and T-tubules of skeletal muscles increase glucose uptake, and the liver releases more glucose during exercise to compensate. Energy sources for mild, moderate, and heavy exercise. During mild exercise, a little bit of plasma glucose is used. Mainly, plasma-free fatty acids are used, and a little bit of muscle triglyceride is used. During moderate exercise, a little bit of plasma glucose is used. About a quarter of plasma-free fatty acids are used, and then a little less than that, than the fatty acids, the muscle triglyceride is used. For about a third, or even more than that. I don't know. It looks, it's hard to describe. Muscle glycogen is used as a source of energy in moderate exercise, and it is the largest source of energy during heavy exercise, muscle glycogen. Muscle triglyceride, plasma-free fatty acids, and plasma glucose are used in small amounts, and they're all used in about equal amounts. They make up about a third of the energy source, while in moderate, those three groups make up about half, a little bit more than half, and then muscle glycogen fills in the rest. Muscle glycogen is not used during mild exercise, and all of that is in the 0 to 30 minute range. In the 90 to 100 minute range, for mild exercise, it is about the same, except more plasma-free fatty acids are used, and just a sliver of muscle triglyceride is used, and about half more than that, or about twice as much of the triglyceride is what is used for plasma glucose. For moderate exercise, we have a little over half of it coming from plasma-free fatty acids and plasma glucose. It's just a little bit of plasma glucose and then quite a bit of plasma-free fatty acids. The other less than half is the sources are muscle glycogen and muscle triglyceride, and they're about equal, but a little bit more glycogen is used. I think the main idea here is that during mild exercise, muscle glycogen is never used as an energy source, and it is an increasing energy source with the intensity of the exercise increases. So muscle glycogen is used during moderate and heavy exercise, but not mild exercise. The main source of energy for mild exercise is plasma-free fatty acids. Energy sources for muscle contraction. ATP is the primary source of energy, but stored ATP in muscle fibers can only power vigorous exercise for a few seconds. New ATP can be produced from phosphocreatine, fermentation, which is not anaerobic respiration, and aerobic respiration. Metabolism of skeletal muscles. The intensity of the exercise can be measured by the VO2 max, or the aerobic capacity, and this is the maximum rate of oxygen consumption by cellular respiration, and it can be increased by training. The lactate threshold, or the anaerobic threshold, is the point during exercise when lactate rises significantly in the blood, and this is typically around 50 to 70 VO2 max. What influences the strength of a muscle contraction? Number one, the number of active motor units, and really this comes down to the number of muscle fibers contracting. Number two, the frequency of stimulus or of stimulation. Number three, the thickness of the muscle fibers, because thicker fibers produce more strength. And number four, the initial length of the muscle fiber. Isometric contractions involve increases in tension but no change in length during contraction. Isometric contractions are used to maintain posture. Isotonic contractions occur when tension on a muscle is not equal to the opposing force, the force opposing the muscle, and this results in changes in muscle length. Isotonic contractions are used to move the skeleton, and there are two types: concentric or shortening contractions. This is when the muscle tension is greater than the force opposing the muscle, and the muscle shortens. For example, when you curl your arm, that's a concentric contraction. Eccentric or lengthening contractions are when the opposing force is greater than the muscle tension. For example, uncurling the arm. Force opposing the muscle is greater than the muscle tension. I believe, yes, eccentric lengthening contraction. This will make the muscle lengthen a bit. And an example of this is when you're uncurling your arm with a weight in it. Stimulus frequency and whole muscle contraction. Action potentials travel or last about 10 milliseconds, while a muscle contraction can last 100 milliseconds. Muscles are always sensitive to stimuli, and stimuli may come faster than a muscle's contraction cycle. High-frequency stimuli may cause a tetanus, and this can be smooth if it's complete tetanus, or jerky if it's incomplete tetanus. But a tetanus is always a sustained contraction. the increased calcium concentrations outside the sarcoplasmic reticulum are important, because if you don't have calcium in the cytoplasm, you can't contract. With incomplete tetanus, there are small instances of relaxation that can be seen depending on the frequency of stimulation, but with complete tetanus, there is no relaxation phase. It kind of plateaus, and then there is some fatigue because the muscle cannot hold that contraction forever. Recruitment is the activation of more motor units, resulting in stronger contractions. Treppe. When athletes warm up, treppe, increased blood flow and temperature help improve efficiency. Summation. Increasing the frequency of stimulation of a muscle gradually increases the force of contraction. Incomplete sequestration of calcium is important because, again, we need that calcium in the cytoplasm to bind to troponin and cause contraction. Stimulus strength and the motor unit response. Muscles respond to stimuli in a graded fashion, and more force is generated for a more demanding task. Force can be increased in two ways: summation, which is increasing the force of contraction of muscle fibers, and recruitment, increasing the number of muscle fibers contracting. A muscle twitch. This is a single contraction and relaxation cycle in a muscle due to an electrical stimulus. This consists of a latent, contraction, and relaxation phase. The latent phase, let's see here. The latent phase is when the shock produces an action potential that is conducted along the sarcolemma and stimulates the release of calcium from the sarcoplasmic reticulum. The contraction is when calcium binds to troponin and stimulates the muscle twitch. Relaxation is once calcium is not bound to troponin and the muscle can relax because it's not excited anymore.